Everyone to our LEC seminar this week. Before we begin, just a reminder that there's going to be a reception with food and drinks and a chance to talk to our speaker after the talk. So please join us for that. And today I would like to welcome Dr. Jacoba Dorley here to give a seminar for us today. He is originally from Liberia and then moved to the US to do an undergrad in petroleum and natural gas engineering at the Pennsylvania State University in 2016. Then stayed there to complete a master's in civil engineering with an emphasis in water resource engineering in 2018. Then in 2023, he earned a PhD, so earlier this year, congratulations, from University of New Mexico in water resource engineering. And he's currently a postdoc at the National Great Rivers Research and Education Center just across the way in East Alton, Illinois, where his research focuses on the effect of road salt application on water quality and aquatic invertebrate drift behavior and survival. So thank you so much for joining us today, and we're really excited to hear your talk. If you have questions, as always, please save them for after the talk, and I'll pass around the mic.
Thanks for the introduction, Stella. It's great. I hope, because I'm an engineer, that did not destroy your memory. I'm not a biologist, so I'm going to try as best to entertain you and kind of make it interesting for us. First, before I go into my talk, my research journey, as Stella mentioned, has been very adventurous. Started up in Pennsylvania, which is not highlighted here. I've been fortunate enough to do research across the Midwest, the Southwest, and Northeast of the US. So I kind of very broad, diverse background in terms of my research. However, today's challenge is not about my adventure, but I actually Google a lot. How can an engineer for this? And so please let me know after the talk if I actually did a good job. And so most of the work I'm going to show today is work that I have led, and some of my collaborators have led as well. And I'm going to first give a big thanks to my PhD advisor, Dr. Ricardo Gonzalez-Pinzon, and a lot of mentors and collaborators over my PhD, and also with a lot of collaborators. As you mentioned, as you imagine, there's a lot of universities and funding institutions that made this work possible. And so big thanks to them. Plus, I would be remiss if I did not recognize all of the undergraduate students that helped me in the lab work, because, you are biologists, you understand that undergraduate students make this work. So first, I'm going to be talking today about stream ecosystem. And I'm not sure if a lot of you are familiar with it, but stream ecosystem is controlled by natural processes. These natural processes include hydrological processes, meteorological processes, and biogeochemical processes. So just a short definition of, when I say hydrological processes, for instance, those are the processes that regulate the distribution of precipitation across space and time, both in the surface and subsurface of streams. Meteorological processes, for instance, regulate the frequencies of these precipitations. And biogeochemical processes, which is where my research focuses on, combines the meteorological processes and hydrological processes to create relationships between chemical resource supply to sustain aquatic food webs. However, over the recent century, anthropogenic sources have been a great, have added a lot of stress to these ecological processes. From agricultural byproducts, for instance, the overuse of fertilizers. We all drive, we want to go to work, and we want to go to the grocery store. That also creates a lot of stress on the environment. We depend on industries to create products that we need, also create a lot of stress. And all of these stresses propagates down to stream and river networks and affect the chemical composition of these river networks and also the aquatic animals within these networks. And these these stresses also in turn propagate to create high implications for climate change phenomena that we're seeing, like flooding and drought, and also effect on our food and water security. And so when we talk about extreme ecosystem, I'm mostly focused on like ecosystem metabolism. And so the long definition from the Encyclopedia of Earth Sciences is that it is the total energy processed by individual organisms within an ecosystem. I like to think of it in the sense that ecosystem metabolism as a function of gross primary production and ecosystem respiration. And as you can see, this metabolism encompasses the entire like energy food web, pretty much just the ecological structure of things. For instance, you have microbial communities which get their energy from like plants. You have like, and then these microbial communities serve as source of energy for microinvertebrates and fishes. And so in a way, like the entire ecosystem is a fractal network and they're all connected. And so that's why I'm going to come to you today from a microbial perspective and not the big fish, because I don't know that. And so, however, there are limitations in terms of how like hydrologists and ecologists measure these like metabolic processes within stream ecosystems. Some of these limitations include like to measure metabolism, it requires high frequency data for dissolved oxygen, wind speed, and all of these things. And so many times, if you're a small university, or for instance, you do not have all of this equipment to to be able to quantify metabolism. There is also the R-squared approach based on oxygen mass balance, which also affect how we measure metabolism. So if you look at a lot of studies looking at ecosystem metabolism, there are a lot of like differences in terms of how people quantify it. And a lot of these differences come mostly from like reaeration coefficients. And also the effect from groundwater, there's no way we're not able to quantify the dilution or the mixing effect from groundwater to surface water. And so the oxygen, if the oxygen level in groundwater is very low compared to that of the stream, there is also bias in terms of how we quantify metabolism. And lastly, because a lot of metabolism occurs in stream ecosystems occur within these highly shallow groundwater and a lot of these small cavities, like the temperature within these cavities in the groundwater is so different compared to the stream. So many times we'll have our sensors within the mainstream network to measure DO, but then we want to use the temperature within the stream to correct for metabolic processes, but these things are also create some biases. And so over the past decade, or I would say maybe about 20 years ago, instead of focusing on metabolism, hydrologists have started to focus more on respiration. How can we quantify respiration? Because it creates more of a standardized process compared to metabolic processes. And so now bring me to my topic of aerobic respiration. What is aerobic respiration? I'm going to give you the long definition first that I just got from Google. But the key word I want you to take from here is that the molecular oxygen of that. So what I mean by molecular oxygen, if we consider like a chemical compound of sorts, right? And let's say this chemical compound undergoes some sort of biological reaction within the system, right? And it transforms into this other compound here, a daughter compound. And the only difference between these two structures here is the oxygen ion, right? And so that removal of the oxygen ion is what we refer to as aerobic respiration within our systems. And so it's very easy to quantify this. And we have over the past 20 years, there have been a lot of papers and a lot of studies that focus on different methods in terms of how to quantify these oxygen. And a lot of this, the removal of this oxygen that I'm going to focus on here is the removal by microbial communities. And so during my PhD, I focused mostly on working on using this smart tracer resizing and rest roofing, which has been used in the biochemical network for quite a while, for over a century. But however, over the last 15, 20 years, it was introduced to the field of hydrology and ecology as a way to quantify aerobic respiration. And a fun fact about this, I'm the grandchild of this process, because my PhD advisor was the one who introduced this to hydrology and ecology at Oregon State. And he worked on it for his PhD, and I worked on it for my PhD, and who knows? I don't know. Yeah. So when we talk about initially, resizing was introduced to hydrology as a way to quantify separation between surface and subsurface processes. However, we soon come to realize that it could also be used as a way of quantifying like how microbial community use oxygen within screen. So if you consider a particular screen, right, there are different processes that occur within it. We have topographical processes, processes that are driven by topographical exchange. We have processes that are driven by turbulence and variables within the screens. We have different pumping rates within the particular systems, like compartments of that screen. And so this within hydrology is very complex in terms of how we can separate these different processes. And so this is why resizing was initially like introduced. However, it has since grown to be used to quantify like these very metabolic processes. And just this as a notion, I think I mentioned before, like a lot of the microbial processes we're going to be talking about, these are like the high zone, like the metabolically active zones within screen. Like a lot of the biological processes that occur do not occur within the screen over here, this main channel. They either occur within the shallow benthic region or within these side storage. And so I'm going to focus on that. And so what is resizing? So resizing is a venozizing dye where we inject. Resizing is blue in color, and that's that chemical structure I showed you earlier. Once it comes in contact with some living organisms, right, like a live cell, it transforms to a daughter compound resolution, which is pink in color here. And this is the two-division structure. But I like to think of resizing as a binary nanoswitch, in a sense that we're injecting zeros into a particular screen, right? We know the amount of mass of zeros that we're injecting. And then once that zero enters within this metabolically active zone, it switches to 1, right? And then if there is a way that we are able to quantify the zeros and ones someplace downstream here, and then just a simple mass balance, we will be able to estimate what type of aeration, like aerobic respiration is occurring within the system. And so compared to the normal way of doing things, this is pretty simple. This is pretty straightforward because it does not just look at one parcel of water that is occurring within the main channel. They look at like the entire system within that particular channel. And so where the zeros that you see here, those are resizing that did not encounter any living organisms, and the ones are those that encounter living organisms. And the differences between zeros and ones could be explained in many ways, because it could be that perhaps the microbial community are not interested in the type of oxygen that is being supplied. It could be that maybe the hydrology or the screen morphology does not allow the resizing to go into these metabolically active zones. And so most of my research that I did focus on how can we quantify physical, chemical, and biological processes. Like, is it because of the hydrology that perhaps the oxygen is not being used? Or is it because of the biology, maybe like the stoichiometry of these organisms, just does not account for them to use the oxygen? And so resizing, again, just a brief history. It was initially discovered in 1871, and it was first applied in 1929 to test bacterial content of milk. And it was first proposed to the field of hydrology in 2008, and this by Roy Haggerty. And that's my PhD advisor's advisor. And so resizing on the goes, again, just to give you a brief idea that, living organisms is very important in terms of the processing rate of resizing. Within sediment, where we have a lot of microbial communities, like microbial activities occurring, we see that the processing rate of resizing is about an hour, right? But then if we go to like screen water or with sediment, we start to see that the processing rate decreases to like a day, and then to a month with like acid-rich screen water, and then with deionized water, we can see a processing rate of over a year. And so it's highly dependent, the transformation from resizing to restoration is highly dependent on microbial community. This is why this technique is so robust in terms of quantifying aerobic respiration within systems. And the kinetic rate again is pretty much, so there have been a lot of work that are put in place to quantify how resizing transformed from different from resizing to rest roofing. And there have been a lot of work. Some of my colleagues have actually ventured into this particular area of resizing. And so in summary, resizing to rest roofing transformation is proportional to oxygen consumption. It can be used as an indicator for cellular metabolic activity. It can inform groundwater surface water interaction through metabolic processes as a proxy. And it can also be used as an indicator for risk-kill metabolic activity. And so one of my colleagues actually did something. So now that I show you that we're injecting zeros and ones, and then one of the questions you might ask is that, okay, how are you accounting for mass balance? Like perhaps there might be other processes that might be using resizing, and you're not getting a full mass that you injected, like substance, for example, right? Could be something. So one of my colleagues, she was actually a visiting scholar in my former lab at the University of New Mexico. I think she's now because of Padova in Italy. She did some work on resizing, asking the question, does the mass balance of RAS and glucose at a microbial scale? And so in this study, what she did was she used 3 different assays to test it. She used like yeast, pretty much just yeast, and then she used river water from the Rio Grande in Albuquerque, and she used the Bacillus subtilis, which is a facultative anaerobes, to see how these different microbial these different species or these different factors affect the processing rate of resizing. And so the idea here was to minimize suction and photo decay, and so she did not do it in the screen. And on mass recovery, investigate patterns independent of specific microbial species. And so what she found again is like pretty much as we expected, she tested this on like different concentration of resizing. Like as you see from the plot, as resizing increases, which is panel A, you see that it resolution increases as well at different at all concentration. And this is just an indication to show you that resizing does transform. It's an inverse relationship between resizing and the zonal compound. And so what she saw is that she saw higher decrease in resizing for yeast compared to river water and other acids that she tested. And so like her work here, she saw that the mass recovery was about 85 to 110%, and 110% is just based on like standard deviation variances, and so that's what you're seeing 100%. But what she concluded is that mass balance of resizing closes at microbial scale, in a sense that we are able to account for 100% of the mass that we inject of resizing, and whatever mass that we do not see is sort of dependent, it's sort of based on suction, like mostly because it's sub to like the soil or something like that. And as you see from the plot here, is pretty much 100% of the recovery that she got. And then recent work that we just published in Bioscience here is we were concerned about, okay, how is microbial respiration controlled by hydrologic exchange and stoichiometric conditions? And the stoichiometric conditions I'm talking about here is, and I should get into that. So the hydrologist now is saying, okay, a lot of hydrologists say that longer residence downscale always leads to higher biological uptake of carbon, nutrients, and oxygen. And so we wanted to test that. Okay, if we have, if we reduce the velocity of a particular water in the stream, and it takes long for that nutrients to pass through a particular section of the stream, are we going to see higher uptake of that particular solute or not? And then stoichiometrically, we use the Rescue ratio. I don't know, I'm not sure if you guys are familiar with it, but this was a ratio that was proposed by Rescue in 1979, where he said for an ideal system to be optimized, you want to have a 1 or 6 carbon to 16 nitrogen and 1 phosphorus. And so we tested this idea here to see how carbon, nitrogen, and phosphorus, because these are key building compounds that we are all made of, and those are how we process things, and see how it affected that. So normally when we do screen injection, and so here this is if we're doing like a mesothelioma experiment in the lab, and this is where we're doing like some experiment within the field. Normally we'll do injection of resistor. We want to inject it with some sort of a conservative tracer. And conservative tracer, and those are tracers that does not undergo any chemical biological transformation within the system. And so in a sense, if we inject, like for instance, chloride, we expect like 10 ppb of chloride, we expect to get 10 ppb out. And so that's a way to correct for maybe a mixing or dilution that might occur within our system. And so for our research, yeah, if we consider a particular system, what we do when we go to the field is we establish an injection location, right? And then we inject, and then we go downstream and collect samples from that injection location. And that's your students there. And so we usually, what I did for my experiment is that we did like a continuous injection where we filled up a top, like a top with like the resistor, and this is the resistor here in color, in the field, and we stir it out. And then we go and we inject it slowly into the screen. But then we go downstream where that we expect that there will be 100%, like it have fully mixed out. And then we collect that. And so this is what we're gonna collect. So what we're injecting is the zeros, and downstream what we're collecting is the zeros and ones. And then we filter them in the field. And then what we get out of that is that for every time we are sampling, and so we get something known as the tracer breakthrough curve. And again here, the blue there is the conservative tracer. No, it's resistor, and then the red is roofing. In our system. And so for my experiment, what I did was we injected a conservative tracer of sodium chloride, and then with a reactive tracer of sodium acetate, sodium nitrate. So sodium acetate was for the carbon in that C, using that rescue ratio of 1:6 to 16:1. And then monopotassium phosphate, and then we inject the resistor in 2 because we wanted to see how it affected that. We injected for 4 hours. In the stream, because the idea is that we wanted to let the stream go to full saturation so that we get almost like a plateau, like, or something like this, almost like a plateau, like concentration. So for here, what we did to test the hydrology, we did 2 sets of injection. I'll call that round 1 and round 2. Wherein we want to see, okay, how does resistor, how does the microbial community react to resistor with stoichiometry, but also as discharge decreases within our particular system. And so round 1 had like high flow conditions, and round 2 have very low flow conditions. And so that includes the hydrology. And I'll come back to this and show you why that was important. And so normally this is going to be the boring part, and it's not entertaining. Once we do that, we get the breakthrough curve, we go down, and then we use the advection dispersion equation. But then we add another part to it, which is the transient storage, all of the interaction between groundwater and surface water, and all of the side storage. And so this is why they are addition up here. So these terms are very. Let's just consider this as the main channel. We have the reference zone. So this equation just represents how we model a system like that. And then from there we calculate the transient storage time scale in terms of how long does a particular parcel of water stays within that metabolically active zone, and then the transformation time scale and how fast the resistor is transforming to roofing. And then we use something known as the Damkola number, which is DA here, to quantify. So the Damkola number quantifies the hydrology versus chemical chemical versus physical processes. And as you see, like the transformation time scale and then the transient time scale. So the transient time scale is the physical, and the transformation represents the chemical of that. And then for the Demkola analysis, what we say Demkola is approximately one. We say there's a balance between supply and demand, and so maximum retention. And so the molecular communities are pretty much happy. And then if it's much less than one, we say the demand is suboptimal, and then so reaction is limited in a sense that there is a lot of solute that is going into this metabolically active zone, but microbial community are not using them. And then if it's much greater than one, we see suboptimal reaction. And so perhaps like, the hydrology is not allowing the solute to be transferred to those metabolically active zones. And so our results here again, and this is going full injection. So we did 4 different injections. Where we injected just nitrogen by itself, we injected nitrogen and carbon using our respiration, nitrogen and phosphorus, and then carbon, nitrogen, and phosphorus. With that, and again, the discharge that you see, as I told you earlier, is decreases within time. And then round 1 even represents that high flow. Round 2 represents like the low flow conditions in that. So what we saw here is that actually explain. So the panel C here represents the transformation of the plasma, how much the retrogrouping, and that itself as an indicator for metabolic processes that occur. And so what we saw was that perhaps there were not really a lot of statistical. And so the idea that with longer residence time of a particular solute should lead to higher biological uptake did not prove to be true within our case here, because they're pretty much similar to each other. And then this here is just pretty much based on VF here is based on this, like just calculating the velocity, how fast the reaction is occurring. And this here is just like per time. What's the difference? So that was just the discharge. So so if you see here, so here this is discharge. The difference is just the discharge. So we wanted to see if, because in round 2, the idea is that when we expected it low from here, that means the study is going to take a long time to move through the system. But a fast flow, so we expected to see higher uptake here. And then we also calculated discharge and velocity within rounds. We did not see a lot of difference with stoichiometry. And so perhaps the stoichiometry did not affect it as we thought. And so one of our, in a paper that we wrote, one of the things we said is that perhaps the ratio of carbon, nitrogen, and phosphorus that we injected, perhaps we did not account for the ambient conditions within the system, and so we did not meet that ratio, or perhaps the screen was already at that ratio, and we injected and we sort of like disturbed the screen, and that's why my microbial community did not use the what was not effective. So initially we were expecting to see higher transformation rate for CMP here, but we did not see that. And then coming back to the Demkola number, right? So one thing we saw was that for round 1, we saw that pretty much it was tricollinear. And so this agrees with what we expected to see, because what happened is that the water was moving so fast, like the rate in which the soil was being transformed within the system was fast that the microbial community could not process that particular solute. And for round 2 here, we saw that, okay, now it's like reaction limited, that perhaps like, a lot of biofilms have been used in the system. And so the microbial communities were using them, but the supply of that particular solute was not sufficient to these metabolically active zones. And so, well, in conclusion, what we saw was that we saw a decrease in discharge and transient storage downscale. But we saw metabolic processes remain pretty much consistent within the system. And this is just a schematic of what we saw within the stream, where round 1 was like high connectivity between these were cavities, and round two was less connectivity between the cavities. And so what we assume is that because the reason why we did not see the difference between round one and round two is we just need connectivity. And let me see. Okay, I think I can go. Another question we had in and this is the work that we did in Colorado and Iowa, was to see how we can compare how the disturb screen, and I say disturb screen in a sense that, like, screen that is exposed to a lot of fertilizer use, and so there's a lot of nutrients being put into that screen. Versus a screen in Colorado where we had more pristine screen, like it was all natural processes that was occurring. And if we look also, one of the other questions that we had is that does morphology play a role in terms of how microbial community might use resuscitant? And so here is the agricultural screen. And we did this in Iowa, and we actually did it in the middle of a cornfield. And it was not, it was not fun, trust me. This was 2019, the summer of 2019, it was hot. Yeah. And Colorado was nice, but one story there, I saw a bear the first time. I was sampling at 12 a.m. in the morning, and I saw a bear. And I just jumped in the truck and I just filmed it. Yes. And so, yeah. So for that, what we did again was that, okay, we saw that there was difference between this, so the agricultural side was more like a script, and it was, I think it was, I don't know if you guys are familiar with screen order. So the agricultural side was a third order screen. No, it was a fourth order screen, and the forestry side, which is the one in Colorado, was the second order screen. So in a sense, the fourth order screen has more discharge, has more flow compared to the second order screen. And so what we saw is that we saw that recycling was proportional to discharge within that particular system, where we saw because of low discharge here, we saw higher processing rate at the Colorado side. And with high discharge, so we want processing rate. However, these could also be a function of what these microbial communities are exposed to, and that's what we bring the question about the disturbed versus the undisturbed sites. And so another thing, we went and did some correlation to that because we thought perhaps what was seen could have some that could help us to better interpret the data, but so far we did not see anything. But the only thing we saw that we really couldn't was the advective time scale. And the advective time scale is just like how fast the water going straight. We're not accounting for mixing. We saw like strong correlation between that. And so this again just tells that morphology is playing a huge role in terms of how microbial community process resides in the system. And actually, there was actually a good paper on one of my colleagues we published back in 2022. She just looked at the physical processes that will occur within those things. She used like conservative traits and just look at the physical processes. And yeah, but if that's something interesting, we can talk more about that. And again, we did similar to the Demcola number within the system. Here again, we saw that the agricultural side was mostly reaction limited, and then the forestry side was more pristine. And again, that explains why we're seeing, because we see that it's right within this one-to-one line, where it's like theoretically balanced between supply and demand. And so the use of fertilizer within a particular system does affect how microbial community responds to certain type of soils. And so in conclusion, one thing we got, like, how does this two work that I just showed you contributed to biogeochemistry, which is, I like to consider myself more of a biogeochemist than an engineer. First, it gives us new insight in physical and biological scaling of nutrients across food systems. It gives a plausible explanation for the lack of consistency between transient storage and in-stream nutrients processing, and nutrients retention in-stream is unpredictable when soil use-specific analysis is performed. So this was the work we did was the first work that actually looked into different solutes like nitrogen, carbon, and phosphorus, and those stoichiometric ratios. A lot of hydrologists, a lot of the work that are out there just focus on one solute like nitrogen, for instance, right? But then this gives a new insight in terms of, okay, perhaps we need to start looking at stoichiometry, and that might affect it. And then we need to start looking also again at, physical processes. And so that was the title of my one of my paper in my grad school, like physical, chemical, and stoichiometric processes in SCREAM. So yeah, and we did publish a few of these in terms of methods. Some of my collaborators and so these are all methods paper that we published over the years in terms of how to use sodium to do these type of experiments, and how biological processes might be used in terms of understanding SCREAM processes. And we also have a video, a YouTube video that we did in 2021, I think 2022 or so, that goes into like the entire idea of like biogeochemistry and biophysics and geophysics and stuff like that. So if you're interested in that, I'll be glad to share that link with you. Thanks.
Hey, thank you for that. Super interesting, especially because it's not at all what I do. Can you talk a little bit about the implications for this in terms of what it's going to mean for agricultural streams, and if we should change their shape, their flow rate? Like, I think everybody in this room probably thinks a lot about agricultural runoff into these streams, and what does this kind of research mean for implications in, should we be rerouting streams or changing the flow rate?
Yeah, one thing I did not mention is that agriculture stream was very straight. Pretty much it was like so we call it a pipe. Pretty much what you're quoting, you just get it back. And so there was not like side cavities or like, much exchange between groundwater and surface water. And so that's why perhaps we're seeing like a lot of like, less transformation of attachment to rest roofing in that system. And so what we could do in that sense is perhaps, what you mentioned, like reroute. And also another thing I forgot to mention is that the agricultural screen that we looked at in Iowa, it was in the middle of the cornfield because Iowa is so flat. So there's a lot of tile drainage. So there was like straight runoff of whatever fertilizer they use. As soon as it rains, it just runs straight into the stream. And so that also affected, some of the results that we saw for that site.
Great talk, thank you. So the question that I had was, in terms of, you discussed the supply-demand balance between like reaction demand and the supply of water.
Mm-hmm.
So what is there an ecological impact to reaching kind of an equilibrium of reaction rate and water supply?
That's a good question. The impact is that, microbial communities, right, are one of the biggest reducer of chemicals within our streams, right? And so once you reach that equilibrium rate, that's when they're most efficient in terms of like, reducing whatever chemicals that we are putting in as humans, right? From just from anthropogenic sources, right? And so the idea is to have the stream reach that level so that microbial communities are efficient in using them. But if they are less efficient, what can happen is that now you have to start asking questions about survivability of a particular species, right? Is there going to be a dominant species that's going to be more receptive to like high concentration of this or less concentration of particular chemical? And so it's that idea. And the goal of hydrologists and ecologists in research like this is to create diversity in the system. And this is work I did not show, but one of my PhD work was we actually went and did some DNA and RNA sequencing of microbial community, and we saw that the bacterial community was not have very less diversity. The highest diversity was with fungi communities, and that also connected and that's why we were seeing some like less reaction going on within a particular system. So it comes down from anything to biology, like diversity. It helps the diversity of the system to just how efficient these macros are to process the chemicals.
I apologize if the question is somewhat naive, but as somebody like way from left field. So you mentioned this is kind of an old technique, some history. So you're pushing the boundary here with trying to understand how respiration rate depends on subtle things like morphology of the stream. Is it well understood how it depends on other things like temperature or something? Do you understand that well? Because especially if you're comparing like a stream in Colorado and Iowa, there's presumably a lot of differences. Do we understand well how others affect?
Yeah, it does. There are actually a couple of papers that found, I think it was from the Linux program, that found that streams that are south of the Mississippi are very great at processing certain type of chemicals compared to streams that are north of the Mississippi. I don't know why that is. It could be because of temperature. It could just be because of just regional location or something like that, how these things process. But suffice I to say, in terms of temperature, another work that we did, one of my colleagues, and I think this is in review in Nature Bioscience. We did like a nighttime and daytime type of injection at the particular stream in New Mexico, where we injected the same amount of mass of nitrogen. This one we only use nitrogen during the day, like midday, when because the dissolved oxygen has a Diurnal circle where the dissolved oxygen is pretty high, and so that idea was looking at mostly gross primary versus like primary production and then respiration. And so we saw very, very significant changes in how this microbial community process between the daytime versus the nighttime. And so that's, that could be one indication for, the type of studies here.
Hi, thank you. Going back to the project about the DNA sequencing for the microbial communities, you said that there was a difference, like a higher versus lower diversity. When sampling, did you take into account like the potential differences at like surface level versus closer to the storage areas that you were mentioning before that have like the higher metabolic interactions?
For that work, what we did, I think I might have an image of that here. That work was mostly a column experiment, something like this. When we went in the stream, and so we went to the stream tallway and collect the sediments, and then actually we collect the sediment and we have a column incubated in the stream for a month, right? And then we brought them to the lab and did the injection of that. So and then this was done across the Rio Grande, like we have multiple sites across the Rio Grande just to understand how it's changing the fluid system. And so it was pretty consistent. And this is the one I'm talking about that we did the DNA sequencing and the sequencing too. So it was not like the normal injection that I talked about earlier.
Do we have any other questions? All right, well, thank you so much, and we'll see you guys across the hall for others.